to my channel. In today's video, I'll be talking about cell death. Just like I mentioned in my previous video, that the most common cause of cell injury is hypoxia and ischemia. There are other causes of cell injury such as physical agents, chemical agents and drugs, infectious agents, immunological reactions, nutritional imbalances, genetic defects, and so on. An injured cell will have a different structure, or the structure of an injured cell will change from what it used to be before to something else. The membrane of the cell might be damaged, which will result in the cell accumulating excess water from the extracellular space and it will become swollen. So the cell can return back to its normal state, the way it was before it was injured, if the stress is removed, and this is called reversible cell injury. But most times, due to persistent injury, the cell will reach to a point of no return and die, and that is cell death, which we'll be talking about today. So cell death is the ultimate result of cell injury, and there are two main patterns of cell death: necrosis and apoptosis. In today's video, we'll be talking about necrosis. Necrosis refers to a sequence of morphological changes that follow cell death in living tissue or an organ. The keyword here is sequence of morphological changes. Sequence of morphological changes. Remember that when I talked about the morphological changes of the reversible cell injury, I mentioned that there is hydropic change, fatty change, hyaline change, and mucoid change. So for the irreversible cell injury, which will result in cell death, the morphologic forms that we'll be talking about here is necrosis and apoptosis. So back to necrosis, I've already mentioned that necrosis refers to a sequence of morphological changes that follow cell death in living tissue or an organ. If you don't remember anything from today's video, just remember that necrosis is a sequence of morphological changes that follow cell death. Now, necrosis results from the action of degradative enzymes on the irreversibly injured cell. Remember that the lysosome, which is a membrane-bound organelle within the cytoplasm of the cell, contains hydrolytic enzymes, and these hydrolytic enzymes become released from the lysosome when the membrane of the lysosome is damaged due to cell injury. Having said that, the morphological appearance of necrosis is as a result of two processes. One, enzymatic digestion of the cell, which is as a result of the digestive enzyme derived from the lysosome of dead cells themselves, that's autolysis, or from the lysosome of infiltrating leukocytes, and that is known as heterolysis. The second process is denaturation of proteins. So the morphological appearance of necrosis is as a result of 1. Enzymatic digestion of the cell, 2. Denaturation of proteins. Now let's look at the etiology of necrosis. Let's not forget that necrosis results from persistent cell injury. Some of the causes of necrosis include poisons which can be from chemicals, example drugs, insecticides, fungicides, phenol, uranium, nitrates, and so on. Also toxins produced by pathogenic organisms. Plant poisons, example from some mushrooms, croton oil, animal poisons from bee stings, parasites, and so on. Ischemia or loss of blood supply produced by thrombus and embolism, or by compression of an artery by tumor or an abscess, can also result in necrosis. Mechanical injuries, thermal changes, electric shock, all of this can result or cause necrosis. So now let's look at the gross appearance of a necrotic area. Remember that gross lesions are those changes in a tissue or an organ that you can see without the aid of a microscope. You can see them just by looking at the organ or tissue. Okay, now necrotic areas appear white, gray, or yellow in color, and the tissue appears cooked and stand out from the surrounding normal tissue. Their borders are surrounded by red zone of inflammatory reaction. That is to say that necrosis initiates inflammatory reaction. Microscopically, when you use a microscope, this one you can't see it just by looking at the organ or tissue. You need to use a microscope. There is increased eosinophilia. That is, there is presence of more eosinophils in the blood, and this will make the tissue to stain more intensely pink with eosin than normal cells. Still on the microscopic lesions, the cell outlines are indistinct most times or absent. The cytoplasm becomes vacuolated, and some changes are also seen in the nucleus, and the nuclear changes appear in the form of three patterns: charolysis, which is nuclear fading; pycnosis, which is nuclear shrinkage; charoesis, which is nuclear fragmentation. The next thing we'll be looking at is types of necrosis. Having known what necrosis is, the processes that are involved in the morphological patterns, and the causes of necrosis, and the appearance of a necrotic area, the next thing that we'll be looking at is the types of necrosis. Okay, once the necrotic cells have undergone these early changes that I just mentioned, the necrotic tissue may show distinctive morphological patterns depending on whether protein denaturation or enzymatic digestion predominates. Earlier, I mentioned that the morphological patterns of necrosis is as a result of two processes. One is enzymatic digestion, and the other one is protein denaturation. So the morphological patterns of a necrotic cell depends on these two processes. So when protein denaturation is the primary pattern, coagulative necrosis develops. When enzymatic digestion is dominant, the result is liquefative necrosis. In special circumstances, caseous necrosis and fat necrosis may develop. So I just mentioned the types of necrosis. We have coagulative necrosis, liquefative necrosis, caseous necrosis, and fat necrosis. Now let's talk about the types of necrosis one after the other. The first one here is the coagulative necrosis, and it is the most common type of necrosis. It is characterized by the preservation of the basic cellular shape. The cellular shape is preserved, then and this will permit the recognition of the cell outlines and the tissue architecture. So when you look at it, you recognize what the cell is. The nucleus is usually lost, and it results mainly from the denaturation of proteins. The main cause of coagulative necrosis is ischemia and hypoxia. Grossly, the necrotic area is firm, dry, and has a homogeneous, opaque, cooked appearance. Microscopically, the architectural outline of the tissue or organ is maintained, but the cellular details are lost. The second type of necrosis we'll be talking about is the liquefactive necrosis. It is characterized by the transformation of the tissue into a liquid mass in which cellular and architectural detail is lost. The cells are completely digested, and it results from the action of the powerful lysosomal enzymes. So it is mainly caused by bacterial infections. Grossly, the necrotic area is liquefied and may be watery, tenacious, or semi-solid in consistency. Microscopically, no architectural or cellular detail is visible in the area of necrosis. The third type of necrosis is the caseous or cheesy necrosis. It is characterized by the conversion of dead tissue into a homogeneous granular mass resembling cheese, as caseous or cheesy. Both cellular detail and architecture are lost, and it is encountered mainly with tuberculosis in animals. Tuberculosis is caused by Mycobacterium. Grossly, the necrotic area is granular, resembling cheese. Microscopically, architectural and cellular details is not seen. So the last type of necrosis that I'm talking about is the fat necrosis. And this is the death of adipose tissue within the living individual. And fat necrosis is of three types. pancreatic fat necrosis, traumatic fat necrosis, and nutritional fat necrosis. Pancreatic fat necrosis is the death of adipose tissue in the vicinity of the pancreas due to the action of lipases, whereas traumatic fat necrosis is the death of adipose tissue in the areas of mechanical injury. Nutritional fat necrosis occurs in starving or depleted animals. And that's it for necrosis. Don't forget that necrosis is a sequence of morphological changes that follows cell death in a living tissue or an organ. And two main processes are involved in the morphological pattern of necrosis, and that is enzymatic digestion or protein denaturation. Necrotic areas appear white, gray, or yellow in color, and the tissue appears cooked and stand out from the surrounding normal tissue, with their borders surrounded by a red zone of inflammatory reaction. Then microscopically, there is nuclear changes which appear in three forms or patterns: karyolysis, pycnosis, karyoresis. The cellular outlines are indistinct or absent, and there are four types of necrosis: the coagulative necrosis, the liquefactive necrosis, gaseous or cheesy necrosis, and fat necrosis. And that is it for our video today. Don't forget to hit that subscribe button and ring the bell icon so you don't miss my future video. And thank you for watching. Please don't forget to leave your